Hello everyone, this is Ashutosh Mishra and today's topic is glycolysis. So we are going to explain the glycolytic pathway, right? Glycolysis, right? So first of all, let's have a look on what is this process and why the name is glycolysis. First of all, the glycolysis is composed of two Greek words, right? Now, one is glyco and another one is lysis, right? What is glyco and what is lysis? Glyco is the word which in Greek means glucose or sugar unit, right now, which means glucose or sugar. Glucose or sugar unit is glyco, right now. And what is lysis? Lysis means it's breaking down. It's breaking down is lysis. So basically, this glycolysis can be defined as the process of lysis of sugar, right? This is lysis of sugar. Lysis of is known as glycolysis. That's okay. So this is lysis of sugar known as glycolysis. Now, who has discovered this glycolytic pathway? First of all, on the basis of that, the glycolysis can be given another name, that is EMP pathway. Right? What is this EMP? This EMP means Amden, Meerhoff, and Partners. The EMP pathway. What is that? So these are the name of the discoverer, that is Amden, Meerhoff, and the partners, and that's why it is named as EMP pathway. Okay, what is the second point regarding this glycolysis? The second point would be where the glycolysis will occur. So it occurs in the cytoplasm of all the cells, right now. So it will occur in the cytoplasm of all the cells, irrespective of the cells, whether this is prokaryotic or eukaryotic. The cell, the glycolysis will occur in the cytoplasm of the cell, right now. Okay, now right after that, let's get to know that at or how many steps it will take to complete the glycolysis. So it's a multi-step process. Multi-step means roughly it's a multi-step process, right? Multi-step process. So multi-step process means roughly it will take 10 steps to complete, right now. So it will take 10 steps to complete it. And what will happen in the glycolysis? Generally, from glycolysis, the sucrose or the glucose molecule will be converted to glucose, or sucrose will be converted to pyruvic acid, right? Pyruvic acid or pyruvate. So this is what going to be happen during the glycolysis pathway. Now let's have a look at how many steps are there and what are the significance of those steps. Right now, so let's start with the sucrose. Okay, so initially our body has sucrose or the sugar molecules. Okay, so it would be sucrose right now. Now that sucrose will be converted to glucose molecule. That glucose in the next step will be converted to glucose 6 phosphate. Be converted to glucose 6 phosphate. From glucose 6 phosphate, another molecule will be formed, and that would be fructose 6. Fructose. From fructose 6 phosphate, another molecule will form, and that is known as fructose 1,6 biphosphate. Fructose 1,6 bisphosphate or biphosphate. Right? Now, from this fructose 1,6 bisphosphate, another molecules will be formed, and that would be called as dihydroxy acetone phosphate, DHAP, and that would be in the dynamic with that is in equilibrium with another component, that is glyceraldehyde 3 glyceraldehyde glyceraldehyde 3 okay, it's clear, fine. What will be the next step? This DHABP will now be converted to 1,3 bisphosphoglycerate 1 bis or 5 phosphoglycerate, right? From this 1,3 bisphosphoglyceric acid, it will form 3 phosphoglyceric acid, phosphoric acid. From 3 phosphoglyceric acid, it will form 2 phosphoglyceric acid, 2 phosphoric acid. From 2 phosphoglyceric acid, it will further form 2 phosphoenol, phosphoenol pyruvic acid, 2 phosphoenol pyruvic acid, and ultimately it will form from 2 phosphoenol pyruvic acid, it will form pyruvic acid, right? Acid, right now. So that's a 10-step process, right now. And just to flourish this 10-step process, at different steps, different enzymes will be used, right now. As in case of sucrose, an enzyme will be used, and that is known as invertase. So an invertase enzyme will take this sugar into, right? And to this glucose molecule, another component will be there, right now. First of all, an enzyme is used up in this process to convert glucose to glucose phosphate, and that enzyme is known as hexokinase. Hexokinase. Along with that, must keep it in your mind that this is the step where one ATP will be utilized, right? One ATP will be utilized. So how this ATP will be utilized? We can say that this ATP, it will be formed from ADP to ATP with inorganic phosphate, right now. So how we can write this? We can write this like we can write this like ATP will be formed from ADP, right? So we can write it like 1 ATP will form ADP plus organic phosphate. So that would be utilized, that would be used up, right now. 1 ATP will be used up here, right? The next step where the glucose 6 phosphate is converted to fructose phosphate. And this is under the effect of an enzyme known as isomerase enzyme, right? So here, isomerase enzyme is what? Right now, isomerase. Further, when it converts fructose 6 phosphate to fructose 1, 6 bisphosphate, here one more enzyme is needed, and that is known as phosphofructokinase. The enzyme is known as phosphofructokinase. And in this step, one more ATP will be consumed. So basically, one further ATP will degrade to form one ADP molecule, and along with that, one inorganic phosphate will be formed in this right now. So because these are the energy requiring process, and that's why one phosphate bond from ATP will be broken down to release the energy, right? Now, the next step where the fructose 1,6 bisphosphate is converted to dihydroxyacetone phosphate. This is under the influence of an enzyme which is known as aldolase enzyme. Aldolase which is converting the fructose 1,6 bisphosphate to dihydroxyacetone. Now, I think two more enzymes which are needed in the process, we have to know about them right now, and we will discuss about them also, right? Okay, so DHAP from conversion to DHAP to 1,3 bisphosphoglyceric acid. One more thing is there, but here nothing is getting synthesized, means nothing is getting used up, but here something is getting synthesized. So what is getting synthesized here? NADH molecule is getting synthesized here, right? So basically from NAD plus and H plus, it is getting synthesized NADH molecule. And how many NADH molecule would be there? It would be two NADH molecule will be formed in this process when converting this DHAP to 1, right? At the same time, when we converting this 1,3-bisphosphoglyceric acid, one more this is the one step where further uh, ATP, direct ATP synthesizing, right now, means by using ADP, adenosine diphosphate, with the inorganic phosphate, it is going to synthesize one more ATP. Actually, one more, not one more ATP, but the count would be around two. So two ATP will be synthesized in this process, right? And usually that step is considered the step where no ATP, nothing is getting synthesized. Generally, 2-phosphoglyceric acid is converted to 3 to 2-phosphoglyceric acid. So only the positional isomerism is occurring here. Now, from 2-phosphoglyceric acid, 2-phosphoenol pyruvic acid is forming here right now. And in this process, one more enzyme is needed, and the name of the enzyme is enzyme enolase. Right? This enolase enzyme is needed to convert this 2-phosphoglyceric acid to 2-phosphoenol pyruvic acid. Right? And here also one water molecule will be released. So this is known for releasing of one water molecule in the process. And ultimately, this phosphoenol pyruvic acid is converted to pyruvic acid by using an enzyme pyruvate kinase. By using enzyme pyruvate kinase. Okay. Now, under the influence of this pyruvate kinase, the 2-phosphoenol pyruvic acid is converted into pyruvic acid, that is 2 moles of pyruvic acid, right? And this is the process again where 2 ATP is getting synthesized, that is from ADP to the presence of ADP and inorganic phosphate. Further, ATP is getting synthesized, and how much ATP? Further, 2 ATP is getting synthesized, right now. This is how we can keep it in mind that how many ATPs are getting synthesized and in what step, what process are really needed right now. Now here, basically we have to think about that how many processes or how many steps where the ATP is consumed, right? So we have to keep it in your mind. In the case of respiratory balance state, will be asked for the step where ATP has been synthesized. So ATP synthesis, right? ATP synthesis occurs in two, sorry, ATP synthesis. ATP synthesis occurs in two processes, means direct ATP synthesis. I'm talking about the direct ATP right now, where direct ATP is formed from ADP, that is substrate level of phosphorylation, right? Okay, and is known as substrate level of phosphorylation. Substrate level phosphorylation. This substrate level. So how many ATP synthesized directly? It is total 4 ATP. Total count would be 4. Count would be 4 ATP. How 4 ATP? Because one, this is the process where it occurs, and this is the second process where the ATPs means formed or synthesized right now. So total 4 ATP, 2 ATP and 2 ATP, total 4 ATP is synthesized in this direct ATP synthesis process right now. So this is the first point. Second point would be How many ATP is consumed? How many direct ATP consumed? Direct ATP consumption. ATP consumption. So direct ATP consumption will be around 2, only 2 ATP. Count would be around 2 ATP only. Count would be 2 ATP. Why 2 ATP? Because the first ATP is this is the A, where the first ATP is getting used up, and this is the process B, where the second ATP is getting used up in this process. So here I'm also writing the steps where the ATP has been formed and released. So this is in the step 1 and step 2, where I have marked it right now. And here it is in step A and step where the 2 ATP has been used up or consumed. Now the third one, where the indirect ATP synthesis occurs, right? Where the indirect ATP synthesis. So what do you mean by indirect ATP synthesis? Here, NADH, that NADH has been formed. This NADH is actually a source of indirect ATP because one NADH is equals to one NADH is equals to NADH is equals to 3 ATP molecules, around 3 ATP molecules, right now, and that's why uh, 2 NADH will around 6 ATP, right now. So total count of indirect ATP would be 6 ATP, as 2 NADHs has been formed now, so 6 ATP, and that is given a name step 3. Just mark it like step 3. The step 3 we have synthesized 2 NADH, that is 6 ATP indirectly, right now. So what is the net gain of ATP process? What is the net gain of in EMP process? Then right now is equals to total gain is 10, that is 3 plus 2 plus 4. Sorry, 3 plus 2 uh, count would be 4 now because direct ATP is 2 times. 2 ATP has could be 4. Okay. So how many ATP has been synthesized here? 4 ATP and 6 ATP. So total 10 ATP. Total 10 ATP has been synthesized both directly and indirectly, minus 2 ATP. Sorry, 4 ATP. How it comes to 4? Direct ATP. Right. We are doing right. It was not 4 ATP. It was 2 ATP because directly 2 ATP has been consumed. Okay. So total 10 ATP because 6 ATP here and 4 ATP here. Total 10 ATP, 10 ATP minus 2 ATP consumption, and that would be equals to 8 ATP net gain. That is the net gain of ATP in glycolysis. Thank you very much for today.